Hi, my name is Madison Darmofal, and I am a fourth-year PhD student in the Tri-Institutional Program for Computational Biology and Medicine. Today I'm going to be talking about my work entitled Genome-Derived Diagnosis, a Deep Learning Model for Tumor Type Prediction with MSK-IMPACT. So in cancer, the primary tumor's organ of origin is the strongest determinant of its clinical treatment regimen, and while this seems like a straightforward problem, if a patient's tumor is poorly differentiated, it can be difficult for a clinician to specifically identify what kind of tumor a patient has. So there are also additionally challenging clinical scenarios, such as distinguishing between a metastatic sample and a second primary sample in patients with a history of cancer, and then cancers of unknown primary, where the tissue of origin for a patient's tumor is unknown and therefore unusable to guide treatment. Related to this, there are many mutations and signatures that are associated with specific cancer types and could inform the tissue of origin. So for example, studies indicate that ABC loss of function mutations are associated with colon cancer. The substitution signature for UV exposure is commonly found in melanoma. And these are just a few examples of what is a large and growing field of genomic alterations that are associated with tumor types. And the presence or absence of such mutations might lead a clinician to change a diagnosis in a particularly challenging case. So this has led several groups to develop machine learning models to try and use this data to predict tumor type. This paper was published by the Morris Lab in 2020, and it provides a prediction across 24 common types. And it's similar to a few other methods in the literature, which essentially use whole genome sequencing data to predict tumor type. And while these models are accurate and successful in this own right, the fact that they rely on whole genome sequencing data is actually problematic because this is not yet widely implementable into a clinical setting due to the cost of such sequencing. So clinical genomic sequencing is often done via a fixed panel array, which profiles commonly found oncogenic driver mutations. But from the literature, the majority of classifiers built on these mutations have very low accuracy. So we need to find a way to adapt these classifiers to allow for predictions that are clinically relevant and implementable. So thankfully, I work at MSK, and at MSK, solid tumor genomic profiling is performed by the MSK-IMPACT assay. It is a hybridization-based panel which sequences 505 genes to about 700x and reports mutations, insertions and deletions, copy number rearrangements, as well as several other features. This data set could help us overcome the performance barrier for driver-based classifiers because of its integration into routine treatment at MSK. So since it was FDA approved in 2017, over 60,000 patients have been sequenced, and we hypothesized that this would enable the development of a classifier that is based off of driver alterations that can be more successful than what is currently seen in the literature. So just to introduce the model and the data set, this is a distribution of the cancer types that are available that we're including in our classification. So there are over 40,000 samples across 38 common types that we are using in our training and testing. The orange indicates a simple version of the model that was previously published, and the blue is the more complicated version that I am presenting on today. There are over 44,000 features derived from MSK-IMPACT. It's mostly binary, so it's a pretty sparse set, but it does cover a broad range of different mutational alterations that could lead to a tumor type prediction. And then lastly, like I said, this more sophisticated model is a neural net ensemble model, similar to the model created by the Morris Lab in 2020. And I just wanted to see if, since that model was successful in that context, if a similar architecture would be successful in this context. So the original random forest classifier model had a 74% accuracy and 66% macro precision, which is type average accuracy. And while this was successful, it is less accurate than most of the whole genome sequencing based methods that we've seen before. When I then expanded to 38 types and changed the architecture as well as some of the features, I saw an increased performance of 80% accuracy and 67% macro precision. And this was encouraging because even though I'm increasing the number of predictable types, I'm seeing a performance gain, which is indicating that the model is successful. This graph shows the comparison of the random forest performance to the neural net performance. And you can see that for all but 2 of the types that were available for classification by the random forest, the performance improves in the neural net. And then an important thing for a clinical implementation is that you are confident in the predictions you are putting out. And so for GDD-RF, we instilled a threshold of 75% confidence, where if it's not above 75% confidence, you don't report the prediction. And so when we look at a similar threshold for the GDD-NN model, we see an over 90% accuracy on 75% of the samples that pass and this 75% of samples that pass the clinical threshold across all 38 predictable types, and a macro precision of 89%. So this is a drastic improvement and similar to the performance of the whole genome sequencing models, which is very encouraging. One other thing that we wanted to make sure was checked when switching from a random forest to a neural net model is that neural nets are frequently confidently incorrect. So we wanted to ensure that it is well calibrated. And from here, you can see that this blue model indicates the calibration of a single neural net, while the orange is the ensemble neural net. And there is a lower estimated calibration error for the ensemble method, which was encouraging. Finally, we implemented Shapley values for prediction-specific feature importance, and we validated these by aggregating them across types and found that this aggregation recovered known gene tumor type associations. So when we aggregate all the Shapley values for colorectal cancer prediction, you see that truncating mutations in APC have high importance, and you see TMPRSS2 fusions in prostate cancer, which is encouraging as well. So I'm going to spend the last few minutes talking about some clinically guided novel implementations that I've been working on after integrating into the GDD-NN model. So while in cases for cancer of unknown primary, you might want to predict across all known types, in most cases you can narrow down what you're predicting across given the clinical history of the patient. So you would take your softmax array, adjust it by any known priors that you have, and then you could report an adjusted output probability and a new prediction if the probability changes after your prediction-specific prior distribution adjustment. And so we evaluated this using different metastatic sites because we hypothesized that these could limit the types that should be predicted. So we don't use biopsy site as a feature within our model, but we can define them within the training set of metastatic samples. And so, for example, for a liver metastasis, you might increase the likelihood of a breast or a colon cancer and decrease the likelihood of less seen types such as ovarian and gallbladder, and then use that to recalibrate any predictions that you make. So again, we just would evaluate this using metastatic site as a prior information. And when we do this using this paper to define 33 common metastatic sites across our training and testing samples, metastatic samples, we see an improvement in accuracy of 3%. And this is encouraging. It indicates that the prior is useful, and it also only showed reversal of 44 types and a correction of 133 samples. Was an out of distribution detection feature. So there are always going to be more potential tumor types in the population than feasible to include in the classifier. And so to attempt to identify these, I used a set of MSK-IMPACT samples classified as other, essentially of types that were not included in the 38 types in training, and attempted to identify them. So I did this in two different ways. First, by just using features of the ensemble model, and second, by creating an out of distribution category as a separate training type. And what I found was that the classifier built on metrics derived without an out of distribution class performs better, indicating that we can detect out of distribution samples without explicitly training on them. And in conclusion, we've shown that GDD-NN is a tumor type classifier built on features derived from MSK-IMPACT that returns well calibrated and accurate predictions across 38 different cancer types. And I showed some novel implementations and that the MSK-IMPACT integration will enhance its clinical utility. Thank you.